All right, TGF-beta. So two left, right? WNT and TGF-beta. Um, so this is boring. Not the lecture, but this picture. So I was thinking TGF-beta superfamily, and I thought, because the Sonic the Hedgehog was so funny, and so that fun, and I thought, oh, family. And I thought of the memes of Vin Diesel. So then I asked ChatGPT to include Vin Diesel, and so this is my alternative cover that ChatGPT gave me. Um, so ChatGPT told me, or Dolly or whatever, that this is Vin Diesel studying TGF beta. There's a lot going on here. He look First of all, it doesn't really look like Vin Diesel. He's just bald, but like he looks mad. So his experiment must have failed or something. Anyways, all right. So let's move on. So TGF beta is, like I said, it's a superfamily. So there are uh, all sorts of proteins. We have the TGF beta family, but then within this, there we also have some activins and some other type of genes, BMPs and things of that nature. So it's kind of like a broad group. But let's go into this SMAD pathway. So one of the reasons it is like a super group is because a lot of these receptors combine multiple ligands, right? And so in this case here, we have these a type 1 and a type 2 receptor for TGF beta, and they form two dimers, right? And so we have this receptor type 2 that forms a dimer with another receptor type 2, but that's still only half of what's needed. Then we have a receptor type 1 that forms another bond with a receptor type 1. So you see we need 4 of these membrane bound proteins. So TGF beta, or a beta-like ligand, in this case, this is like part A is kind of the general version. So what happens is there are these serines or tyrosines that are on here that will be phosphorylated once this, just like all of our membrane bound receptors, that generally happens. And this GS box gets phosphorylated, and that causes SMAD to be phosphorylated and then have some action. So that's the very general version of it, right? If we look at part B, these, whatever the ligand is, the different ligands cause different activations and different conformational changes, and therefore different outcomes, right? So in this case, we have activin, nodal, or TGF beta, and that will cause the activation of SMAD2/3, which gets phosphorylated, and then recruits SMAD4, which helps import it into the nucleus and then transcribe this gene. However, if that same complex, same receptors 1 and 2, gets bound to a BMP ligand instead of one of these three, we still have phosphorylation. However, the conformational change is different based on what ligand is there, and the resulting SMAD, in this case SMAD1/5, instead gets phosphorylated. It's still recruited by SMAD4 because that's required to help bring that recruit it into the nucleus or move it into the nucleus. But then we get transcription of a different gene, because this is a transcription factor, SMAD1/5, will bind to a promoter of a different gene. So that's why it's a superfamily, or part of the reason it's a superfamily is that they combine different types of receptors, and these all fall within this TGF-beta superfamily. But depending on which ligand binds these receptors are essentially generalists. Depending on which TGF-beta ligand binds it causes changes which downstream proteins phosphorylated and activated, and therefore what genes expressed. So this is a quick one, example of TGF-beta, and we'll finish up with WNT, and so I will catch you in the next one.